Hello everyone, welcome to Lab 6. Last week I introduced you to a group of gram-negative rods known as the psychrotrophs, which are able to survive in low temperatures, but today I'm going to be talking about a different group of gram-negative rods known as coliforms. But real fast from last week, we talked a little bit about P. fluorescens. So this is a picture right here of how P. fluorescens look under UV. So you can see the colonies are fluorescing, or they're fluorescent underneath the UV light. So I just wanted to give you a picture of that as a quick introduction of what we talked about last week. And in the lab we would have been taking a look at the plates. Unfortunately, it's not something we can do, but let's continue on with coliforms, Lab 6. So for gram-negative rods, last week we talked about the psychrotrophs, which are over here, positive oxidase test. Now, what we're going to be talking about is mostly the Enterobacteriaceae family, which is approximately 50 genera, but not all of them are going to be of intestinal origin, as we'll see in a second. So, gram-negative rods are indicators of spoilage organisms, intestinal pathogens, and then also fecal contaminants, which are going to be coliforms. So, coliforms are all going to be members of the Enterobacteriaceae family, and they're going to be a major characteristic is that they have gram-negative rods, obviously, but then they also are going to ferment lactose to acid with gas. And this is going to happen at either 32 or 35 degrees Celsius. And we have two different origins for coliforms. We have soil origin and intestinal origin. So these are two different origins. It's very important because for our food, we do not want any fecal contaminants. And so these intestinal origin coliforms, such as E. coli, these are going to indicate that there's possible fecal contaminants in our food. So there's fecal matter in our food, and these intestinal origin fecal coliforms are good indicators that this could potentially be in our food. So we want to be testing for any fecal coliforms in the food in order to determine the presence of any fecal contaminants. But we can also find Enterobacter, which is going to have a soil origin, which is not something that we're going to be as concerned about, and it's going to be pretty prevalent, especially in vegetables, or anything that's grown in the soil, right? Fruits, vegetables, anything outside. And then also animals are going to be outside as well in the soil. So we're going to be getting a lot of Enterobacter, but that's not something that we need to be concerned about in our food. The one that we want to be concerned about is E. coli, because that is a fecal coliform, and we do not want any feces inside of our food for obvious reasons. Well, when we want to enumerate coliforms, according to BAM, we have three different techniques. We have the solid medium plating, which would be on VRBA plate. Then we have our most probable number, our MPN method, which has a methodology here, but you don't need to know this. It's not going to be tested on or anything. And then we have membrane filtration as well, which is going to be used for water, if you're going to, or testing any liquids for coliforms. So the first method, VRBA, this is kind of what the plate looks like. It is a dark red color. And then we have these dark red colonies that indicate coliforms, and if they're at least half of a millimeter. And VRBA stands for violet red bile agar, and this is going to be used to detect coliforms because it selects against gram-positive bacteria because of the crystal violet and bile salts. So gram-positive bacteria are not what we want to be testing for because, as we were just talking about, coliforms are gram-negative bacteria. So if we have a plate that selects only for gram-negative bacteria, then we know that we're not going to be growing the things that we're not worried about. So crystal violet and bile salts are important for this selective property of the plate. And then lactose is going to select specifically for lactose fermenters, which, as we saw, is also a characteristic. And then the neutral red, this is going to turn red when acid is produced. This is going to create the dark red color on the colonies because the lactose fermentation is going to create acid, which will cause any coliforms because they can ferment the lactose to turn dark red. So that's how we know that we have any coliforms present on our VRBA. The next method, which is based on the NPN method that we would be testing in the lab, is our SimPlate method. So this can count both coliforms and fecal coliforms. So that's E. coli and the Enterobacter. But we want to detect... I told you that we're mostly worried about the fecal coliforms because those are the ones that indicate a fecal contamination. So how we test for this is through an enzyme known as beta-glucuronidase, which is we're going to be testing for this enzyme, which is mainly found in E. coli, which, as we know, is a fecal contaminant, fecal coliform. So we're going to be using this MUG protein. This is an indicator, and it's a fluorogenic compound that's going to be generating a blue fluorescence when in the presence of beta-glucuronidase, which is found in E. coli. So we just shine our wells with a UV light. We have these positives. These all indicate coliforms. All the red ones are coliforms. And then the ones that light up under UV are E. coli because they have the beta-glucuronidase. So we can see how many coliforms we have based off of the red, and then we can see how many E. coli specifically based off of how many fluoresce under UV. Then our next is the membrane filtration, which would be on MI agar, which is going to detect and enumerate E. coli and total coliforms in drinking water. And this is this is using the membrane filter, so the water goes through the filter. The filter is small enough that it can stop any of the bacteria from going through, and it allows the water to go through. So once the bacteria have been stopped on there, any coliforms, and especially E. coli, will be growing on there. And the beta-glucuronidase is again going to be detected. So it's going to be detecting two different bacterial enzymes, beta-galactosidase and beta-glucuronidase. And it'll get a fluorescent color as well, okay? Fluorogen and chromogen. So fluorogen is for color, is for the fluorescence, and then chromogen is for the color. But you don't have to worry about any of the specifics too much for this. We're not going to do this in the lab or anything. Then we have our IMVIC tests, which are going to be used for distinguishing between fecal and non-fecal coliforms. Because as we were saying, that E. coli is our fecal coliform, whereas Enterobacter, specifically Aerogenes in this case, is our non-fecal coliform. And so when we do the IMVIC test, which stands for indole, methyl red, which is the M, V is the Voges-Prosker, and then the C is the citrate utilization. So each one of these tests is going to be testing for a different thing, and then based off the 4 results for your tests, we can determine whether it's E. coli, which is fecal, or Enterobacter, which is non-fecal. So the first one is indole, which is going to be testing for the breakdown of tryptophan to indole using these medium reagents, and then if it's positive, you're going to get a red ring around the very top. So this is for the indole test. And so the MRVP test, which is for methyl red, MR, and the Voges-Prosker, which is VP. So the MR test is going to be testing for the fermentation of glucose into mixed organic acids. And then the Voges-Prosker tests for the production of 2,3-butanediol. So this is going to be taking place in E. aerogenes. This will be taking place in E. coli. And when we get these tests, we see that the positive result for the MR, the broth is red, and the negative result, the broth has not changed color. It's still kind of this opaque color. And then for the VP, we have this ring on the top, just in the very top of the test tube in the broth is the positive result, and then the negative result is no color change again. So it's very easy to see the difference between the positive and the negative for these ones. It's just whether it's red or not. Red or not. The very last test is for citrate utilization, which is just testing for the organism's ability to use citrate as a carbon source. And so it's on the Simmons citrate slant is the medium, and then you just streak the organism onto this slant. Then when it grows, if it utilizes citrate, then it will turn blue and change from green to blue. If it does not utilize citrate, then it's negative, and the slant will stay the same color. So it's going to just stay green the whole time. Whereas if it does utilize citrate, then it'll change from green into this blue color and be positive. So if we were in the lab, we would have been doing these things, testing everything, but since we can't, just take a look at all these procedures, make sure you understand why we did that, or just the steps that took place at the very least. And if you have any questions, feel free to let me know. Email me, take a look at the blackboard, and I'll see you next week. Good luck.